Hello, in this video I'm going to help you get started on this karyotype activity. So the first thing you need to do, if you haven't done it, is download the worksheet, and the worksheet looks like this. And what we're going to do is go through the activities and quickly show you how to do this first one, and then from there hopefully you can do the rest. So let's get started on this. So you go to the karyotyping activity. And I'm going to just read this real quick to bring up some information that you'll need. So if you've read this already, just listen to me as I do it again and explain things. This exercise is a simulation in human karyotyping using digital images of chromosomes from actual human genetic studies. You'll be arranging chromosomes into completed karyotype and interpreting your findings, just as if you were working in a genetic analysis program at a hospital or clinic. Karyotype analysis are performed over 400,000 times per year in the US and Canada. Imagine that you are performing these analyses for real people that your conclusions would drastically affect their lives. So we're going to show you what a karyotype is in a second. So let's talk about G banding. During mitosis, the 23 pairs of human chromosomes condense and are visible with a light microscope. A karyotype analysis usually involves blocking cells mitosis and staining the condensed chromosomes with gamsa dye. The dye contains regions of chromosomes that are rich in the base pairs adenine and thymine. So you already know this, is that there are 4 base pairs: adenine, thymine, guanine, and cytosine. So this dye only really affects areas of adenine and thymine with a dark band. A common misconception is that all the bands represent single genes, but in fact the thinnest bands contain over 1 million base pairs. That's adenine, guanine, cytosine, and thymine. So 1 million base pairs and potentially hundreds of genes. For example, the size of one small band is about equal to the entire genetic information of a bacterium. So these are the genes, and this is what a karyotype would look like when it's uncategorized. It's just a picture of all of your genes, and there are 2 pairs of each gene. So one, and then there'd be another one that looks exactly like it or very close, because you're getting 1 pair from mom and 1 pair from dad. So 23 genes from mom, 23 genes from dad. That's 46 genes altogether. And the G bands are these bands here, and that's where there's a heavy region of adenine and thymine, and you can use that to help identify the chromosome. So there's 3 things we're going to be looking at: the length of the chromosome, where the centromere is, and the banding to help identify and match up the chromosomes. This analysis involves comparing chromosomes for their length, the placement of centromeres, areas where the 2 chromosomes are joined, right here, the centromere, and the location and size of the G bands, so how big it is and where these G bands are. You will electronically complete the karyotype for 3 individuals. Look for abnormalities that could explain this phenotype. So if we go back to our list, here are the 3 individuals. You'll have patient A, patient B, and patient C, and you'll be answering questions after you do their karyotype. Your assignment. This exercise is designed as an introduction to genetic studies on humans. Karyotyping is one of many techniques that will allow us to look for several thousand possible genetic diseases. You will evaluate 3 patients' case histories, complete their karyotypes, and diagnose any missing or extra chromosomes. Then you'll conduct research on the internet to find a website that covers some aspects of human genetics. If this is an assignment for a class, again this is, and you'll just be turning in this document to Canvas. Okay, so let's go ahead. Let's go ahead and look at these patient histories. So what you do is you click on patient histories, and then we are going to be doing patient A. So patient A is a nearly full-term fetus, so it's a not even born yet, of a 40-year-old female. Chromosomes were obtained from fetal epithelial cells acquired through amniocentesis. So they reached in through the womb and grabbed some skin cells of this fetus, which is a fairly dangerous process, but fairly commonly done. If you need to know genetic history or genetic ideas about your unborn child. And again, if you're 40 or older, we tend to do this just to let you know if you're having someone who has a genetic anomaly in them. So let's click on this, and here's our karyotype. So what you're going to do is just match these up. Place this chromosome in the partially completed karyotype below by clicking on its homologous chromosome. If you match the chromosome correctly, you will proceed to the next chromosome. If you match incorrectly, a page will explain why the chromosome you chose is not the unknown pair, and you can choose again. So all the blue spots are the ones you're trying to match up. So looking at this chromosome, where does it match best? 2, 5, 12, or 16? All right, well, if you look at the size of this, it can't be this size, so it can't be that. Might be 5, might be 2. So we'll go ahead and pick 2. And if it's right, it'll tell you correct. Now click on the chromosome in the karyotype that is homologous to this chromosome. So now we have to match this one up. So it's really small. So probably not that one. Probably not that one. Let's try 16. Correct. All right. So now we have to match this one up. Okay. So too big, too small for that one. Could be that one, and since that's the only choice, and the band here is kind of high up and there's 2 bands above it, 3 below it, so it's probably this one. It's just not a great image because they're not exactly identical, but the banding matches. And then finally we're on our last one, and that looks like it's about the right size. So now you have paired up all the chromosomes. Where does this extra chromosome belong? Because we have all these chromosomes banded up, so there's 46 chromosomes here. But there's an extra one that this fetus had. So where does it go? 13, 18, or 21. So if you click on this one, this unknown is much shorter in length than chromosome 13 in the banding patterns and centromeres. So we know it can't be 13 because it's too small. So let's try again. And it's probably not going to be that so let's try this one. And there we go. So now you have it figured out, and you go ahead and you interpret. So lab technicians compile karyotypes and then use a specific notation to characterize the karyotype. This notation includes the total number of chromosomes, the sex chromosomes, and any extra missing autosomal chromosomes. So the autosomes are all the chromosomes that are not sex chromosomes. So if we look at our karyotype, which is down here, so this is the karyotype. Again, this is all the chromosomes. So there are 46 of them, all right? There are 44 autosomes, 1 to 22 are the autosomes, and the last pair, 23, are the sex chromosomes. So they say there's 44 autosomes and 1 pair or 2 sex chromosomes. So going back to our description, this notation includes the total number of chromosomes, the sex chromosomes, and any extra or missing autosomes. For example, 47XY plus 18 indicates that the patient has 47 chromosomes. So not 46, they're abnormal. They're a male because it's XY. XX would be a female, so they're male. And the extra chromosome is at the 18th. So they have 1 extra 18. It's a male with an extra autosomal chromosome 18. So this person, 46, they're normal. XX says that they're female, so it's a female with a total number of chromosomes. So they had 46. 47XXY means that they have an extra chromosome, and they're male, but they have an extra sex chromosome. It's a patient with an extra sex chromosome. So in your notation, you'll just be picking what it is, and you'll be highlighting the correct answer here. Right? So let's look at that, and then I'm going to turn this off and let you go to work on this. So what you would do then is you would go down and you would see where is it. So these guys have 47 chromosomes because there's 1 extra, and they are XX. If it was XY, this chromosome would be really small. So X is big, Y is a really tiny chromosome, so they are female. And so it's 47 XX female, and then you have to tell me where the third, which of these chromosomes has 3, right? And then you would go to making a diagnosis, and you would just read this and tell me which of these things it would fit, and you would highlight that name. All right, well, hopefully this helps. If you need additional information, please reach out to me, your science teacher.